Welcome back to Bio 111. This is Chapter 18, Evolution and the Origin of Species. We have three sections. Section 1 is Understanding Evolution. Section 2 is Formation of a New Species. And Section 3 is Reconnection and Speciation. All right, so famous scientist Charles Darwin, the view of life and how it got to the way that it is today can be summed up by saying descent with modification. So each generation is slightly different than the previous generation, and some of these changes are selected for, and some of them are selected against, and some of them are lost. And the summation of these changes are what we call evolution. All right, so evolution is a heritable change in one or more characteristics of a population from one generation to the next. And it's famously said that nothing in biology makes sense except in the light of evolution. And the evolution has been called the connective tissue of biology. If you've studied physics, you study chemistry, you know that there's typically some underlying principles that help you understand and connect all the concepts in those fields. Biology really doesn't have that unless you have the concept of evolution there. So evolution really was able to bring this subject together in that way. All right, so when we talk about evolution, we're going to be talking about populations a lot. So what do we mean by population? That's a group of organisms in the same species that live in the same geographic area and have the same opportunity to interbreed. All right, so that's what we're going to be, the definition we'll be using when we talk about population. All right, so some ideas here. Evolution is a pattern of change revealed by data across disciplines, looking at basically a lot of data from different areas of biology. It's a process consisting of mechanisms that produce the observed pattern of change. All right, so we go back and look at fossil record, and we look at the various the diversity of life, and we see a pattern of change throughout it. Evolution is the mechanisms that explain and connect those changes. All right, so kind of have to go back and look at the world before the concept of evolution arose, and that's basically that life. You had this Aristotle's view of the world. Life forms are arranged in increasing complexity. Each is perfect and unchangeable. And then you had the Linnaean classification, how they originally, how Linnaeus originally classified species. We still use this nomenclature system today, but organisms were grouped based on a pattern of creation, so how they look together. So you might have things in families and grouped in that such way. We still use the Linnaean nomenclature. However, we typically classify things now based off of genetics, molecular evidence. All right, so looking at the time that Darwin was developing his views on the world, largely what we see is there is a focus on the age of the Earth and age of the universe is becoming larger than what was previously believed. George Buffon suggested that the Earth was much older than previously believed based off of geologic evidence. George Cuvier, documenting fossil discovery, showed that extinction had occurred, so things could die that kind of lends itself to think that there might be something like natural selection, right? If something like the dinosaurs died, then where did the species that came after these lost species, where do they come from? Jean-Baptiste Lamarck, he suggested kind of an alternative view of evolution, but fundamentally it was that species might change over time. That's still a revolutionary idea. And Charles Lyell argued that geological forces had gradually shaped the Earth and continue to do so. So the idea the Earth is still forming, it was new to that time. All right, so we had fossil evidence that of things like mastodon and giant ground sloth and the Irish elk. If you want to see, just side note, if you want to see a real skeleton, or at least a partial skeleton of a giant ground sloth, the North Carolina Museum of Natural History has one on display. Great place to go look. All right, so fossils, they're remains or traces of organisms from the past. They're typically found in layers of sedimentary rock. You think about why it's found in sedimentary rock. The animal lays down, or the organism lays down, and then soil or other sediment covers it and continues to do so until it's completely covered and is preserved, right? And then paleontology is, of course, it's a study of fossils. If you've watched Jurassic Park, you've seen some paleontologists doing some work. And this is the idea of how the formation of fossils might occur. Sediment are laid out in layers, and anything that might get stuck in these layers is possibly preserved, and that is the fossils that we are finding. The fossils, in some cases, could be, typically fossils are mineralized organisms. So when you're picking up these things, they're not actually organic structures. They're minerals, they're rocks that have formed in the patterns left by the organisms that came before. All right, Lamarck mentioned that he had a kind of an alternative version or early version of evolution. His idea of evolution is kind of silly when we look at it from our perspective today, but at the time it was revolutionary. Basically, he would have explained, such as this picture here, that a giraffe got its long neck because it stretched it out trying to get to leaves, and so one generation would stretch it, and the next generation would stretch it a little bit more, and they'd stretch it, trying to get those leaves, and eventually they'd get to the point where they get the leaves. That's not quite what's not at all how the modern theory of evolution theorizes that those sort of changes occur, but it was an early idea that life was changing, and it's important for that part. All right, so geology really began to realize that some of the changes that were being demonstrated by geology had to occur over a period of time much longer than 6,000 years, and so the idea that the Earth was older than this was important for things for Charles Darwin, because he needed that to explain the long periods of time it took for evolution to occur on. So it changed the time scale that we're talking about. All right, so we got an economist in here. So, part of this is kind of the zeitgeist, all right? So there's the spirit of the times. Things were, the concepts were previously held were being changed. But in this case, we're looking at this idea that populations grow much faster than food can supply them. And only a fraction of the population will survive and reproduce. And this is an interesting idea. It indicates that there are certain groups within populations, there are individuals that will survive, individuals that will not survive. There are fit individuals and there are unfit individuals, and um, perhaps after several generations of this, there'll be more fit individuals than there are unfit individuals, and that's kind of the importance of this concept here. All right, so Charles Darwin, the man of the hour. This will probably be an hour-long video, so it's an appropriate name. He's born in 1809 to a wealthy family. He dropped out of med school. He studied theology at Cambridge. He had a passion for the study of nature. All right, so as a young man, he joined a voyage on the HMS Beagle. He wasn't necessarily commissioned to do this, but he went on this trip with his ship, and he got to see quite a few different areas, and particularly, he famously went to the Galapagos Islands, where he was able to look at some organisms that had been in isolation for many years. And he's famous for his study of finches, right? So he noticed two important patterns. Varied traits exhibited by close related species. All right, so you might look at his finches study. He's famous for his finches. Particularly, these finches had different beak sizes. If you look at these pictures here, we've got, you can tell these three finches have different beaks. And it turns out that these beaks are suited for the particular food, or where these birds. It hinted at that there was some adaptation to their location, that there was some selection for a more fit beak size. All right. All right, and there were similarities between the fossils of extinct species and living species in the same area. So it kind of gives the idea, like our giant sloth, right, that we talked about earlier. We have a small sloth still around. So it gives the idea that there has been change, that there was a larger version of this, or a smaller version of this, or a different version of this, that it's no longer here, but there is an organism with similar traits that did survive. Gives an idea of, again, of survival of the fittest. All right, so we typically, we talk about Charles Darwin a lot, but there was one other guy, that another biologist, who was also working on evolution, and he should get the credit he deserves. So Wallace didn't work with Darwin, but he also at the same time was developing a similar concept of evolution by natural selection. Right, so we need to give him his some credit there. All right, so Darwin wrote a book titled "The Origin of Species." And it detailed some broad observations, unity of life, the diversity of life, and the match between organisms and their environment, right? Okay, so just looking at the shift of the worldviews before and after the publishing of Origin of Species. All right, so evolution can occur due to heritable traits, genetic variation. Now, Darwin wouldn't have known anything about genes and that sort of thing at this time. Remember, Mendel was working on his work in the late 1800s, and it wasn't rediscovered until the early 1900s, and we didn't even know what the structure of DNA was until the 1950s. So genetics is very new, um, but children typically look a lot like their parents, so something's being passed down there. So we knew that. Natural selection, and that's traits that favor reproductive success and survival become more prevalent in a population. Um, you know, one easy thing, the concept to think about when we talk about natural selection is really what is the bare minimum to get to reproduction, right? If an organism can pass its genes to the next generation, it was successful, right? It was fit enough to survive. Um, and then it's on to that next generation to carry the torch to the next generation. All right, so one key requirement for evolution to occur is there has to be some variation in the population, right? We're talking about natural selection. If everything's exactly the same, there's nothing to select for. You can't pick the better out of a group or the more fit out of a group if the entire group is completely uniform. So some variation is required. And also you have to have plenty to select from. You have to have an overproduction of offspring. If only one individual is fit, that's going to be a problem, right? Because you need a mating pair in most cases. And like I said, the main thing here is surviving to pass your genes to the next generation. That is the goalpost here. You've got to survive to reproduce. And so an example is slow rabbits will get eaten by predators more often, and only fast rabbits will survive long enough to reproduce, right? So the fast rabbits will pass their genes on to the next generation. And the key point here is their genes likely hold some of the reason for why they were faster. Adaptation, organisms in a population become better matched to their current environment through natural selection. So over many, many years, many, many generations, as more and more generations are selected, the traits that are better suited become more pronounced. So you think about a porcupine. Those are hair-like projections. They're hair, right? But this particular organism has evolved large quills to prevent it from being eaten by these two lions. All right, so let's go back to our finches. We're going to talk about how evolution might happen. So the first step with these finches. So you have a small population of birds. They arrive on an island. Somehow they're on this island, and they're feeding on seeds of different sizes. Now, something I want you to notice in this picture, this small population of birds, they have different beak sizes, right? Perhaps not that important where they came from, but maybe it might become important moving forward in where they now live, all right, this new island. All right, so these birds produce offspring that also vary in beak size. So, and this variation is due to mutations, right? We have different eye colors in humans, and we have different hand sizes, different hair colors. So these are largely variations without really any selective pressure making go one way or the other. But we're looking at this particular one. Maybe there is some selection that might occur against these. All right, so resources are limited, and not all offspring survive. The seeds are large, right? So some of these birds have really small beaks, so they can't eat these seeds. And what you find is that the birds with larger beaks, maybe they can crack the seeds open. Maybe they can just actually fit the seeds in their mouths. Are actually able to survive. So we begin to see the population shifting towards there being more and more peaks finches with large beaks, and then we begin to see the beaks beginning to become larger still. It's after many generations, there's a new species of bird with larger beaks, as well as other modified traits suited to the island environment. Right, so maybe you see the coat colors changing, that sort of thing. So it's not just a single trait that's being selected against here. All right, so natural selection summary. Natural selection is a process through which evolution occurs. Natural selection can increase the match between organisms and their environment. The environment is always changing. No adaptation is perfect, right? Key thing here, individuals do not evolve, populations evolve. You might be tempted to think, like Lamarck, that an individual is able to change itself over the course of its life, but that's not what we're talking about. What we're talking about, slowly, from one generation to the next, there's a trait that gets picked up, it survives and moves on to the next one, right? You have to see it for over the course of many, many years before you can actually tell that there's a change occurring. All right, so what is some evidence for evolution? Artificial selection. We have some direct observations from the field in the lab. Comparative autonomy, so homologous structures, the fossil record, and then biogeography. So artificial selection, this is where we have essentially taken our hand and forced organisms we've basically bred plants and animals to do what we want to. So kale, Brussels sprouts, cabbage, broccoli, kohlrabi, mustard greens, and collards are all related. They're all in the family of Brassicaceae. And but they all came from a wild ancestor, wild mustard. If you get into doing any biology research in lab, any plant biology research in lab, you'll end up very likely working with something called Arabidopsis thaliana. That is a weed found in Europe, but it's also in the Brassicaceae family. We as a species, humans, have bred this particular family of plants into many, many different types of edible plants, like we said, kale, Brussels sprouts, cabbage, broccoli, kohlrabi. How's the example of or evidence for evolution? We're basically forcing evolution on. These plants have adapted to pressures, to selective pressures we put on them. We only pass on the seeds. We only keep the seeds, or we only plant the ones that we like, right? So we're selecting them. So only the ones we like survive. So it's a type of forced evolution. All right, so one example of kind of where we've seen firsthand evidence of evolution would be in things like bacteria. I said that it takes many, many years to see evolution occur, but I really should have said it takes many, many generations. And in large organisms like humans and whales and cows and whatnot, a generation can be decades. But bacteria, they might divide every 20 hours or so. That means we can get through a lot of generations in a very short period of time. And so you see this graph here, an increase of MRSA in hospitals, right, from 93 to 2005. What we're seeing is that they're building up a resistance to antibiotics, which is a scary thing for us, because we rely on antibiotics to kill these bacteria and to keep them in control and keep them from killing us. But they're developing resistance to antibiotics, and seeing why is this happening. Well, so we take an antibiotic, right? So we're applying a selective pressure to these bacteria. The ones that are able to survive, they survive. The ones that die, they die, right? Let's say that we have one bacterium that happens to have a mutation that allows it to live a little bit longer in the presence, or able to resist a little bit more antibiotic. All others die and it survives. Well, it divides and it starts dividing and reproducing. So now you have a whole population of slightly more resistant MRSA bacteria. As you keep doing this over and over and over again for over a couple of years, you begin seeing much more resistance. So certain strains of Staphylococcus aureus are now resistant to most antibiotics, including the powerful methicillin. Bacteria with a variation allowed them to synthesize cell walls of different proteins, not affected by that antibiotic, and they're able to survive and reproduce. Now, bacteria have another trick up their sleeve. They can swap genes via horizontal gene transfer. If you recall, in a previous chapter, we talked about plasmids, and bacteria can actually hand over these plasmids to other bacteria. And a lot of times these plasmids contain antibiotic resistance. So one population of bacteria may not have been resistant to antibiotic, but they can gain it, which is remarkable. And if you recall from chapter 17, that's actually very useful for us in biotechnology, because we actually use plasmids to culture bacteria to make things like human insulin, and we might be able to use it to make things like synthetic spider silk and things like that. So it's pretty cool, but it also provides some challenges to the medical world. All right, another one is homologous structures. We have this picture here. We see a human arm, a cat arm, a whale arm. I'm going to use the word arm here, or hand, and bat wing or arm. We've color-coded the bones that are homologous. When we mean homologous, we mean these are essentially the same structures in the different species. All right. And it just so happens, if you look at these various organisms, they have very similar bones. They may have changed in length and morphology a little bit, but their arrangement in respect to each other has remained the same largely. Cats and dogs, by the way, they walk on their tippy toes. So their entire foot is actually, their heel is that second bend. You may think that a cat or a dog their rear leg has a reverse knee or something like that, but they really don't. That's actually their ankle. That's their heel. That you see that reversed that reversed knee. They have a higher up knee that's analogous to our knee. So the idea here is that these are structures that come from a common ancestor and have evolved and diverged to look as we see here. So we've evolved a hand that allows us to grip. Cats are able to walk and climb. Whales can swim, so they've got kind of a flipper. You notice inside of that flipper, they don't have separated fingers anymore, but digits anymore, but they still have the bone structures inside there that are analogous to fingers. And then bats, you can see their arm and hand have adapted to allow them to fly. It's a function as a wing. All right, so another example of this is looking at embryos. Look at this chick embryo, look at this human embryo. They're very similar to each other. It's only as you start getting further along in development that you begin to see structures that begin to differ, that you can change. You can tell the difference between something like a chick embryo and a human embryo, and these are fairly unrelated organisms, right? You might not be surprised if you saw a monkey embryo and a human embryo, or a chimp embryo and a human embryo, but this is a chick. This is a chicken compared to a human, and they're remarkably similar. Um, the idea being that the process of development are largely inherited or largely unchanged through evolution. All right, there's also vestigial organs. These are features that have no current function, but they resemble structures of ancestors, and we can point to homologous structures on other living organisms. So, look at the manatees. They have fingernails. They don't have a need for these fingernails. They're pretty much useless. They're just kind of like patches of fingernail material, but they're there nonetheless. And if you've ever seen an elephant's foot, they look remarkably similar to an elephant's foot to their fingernails. And they're close. Manatees are close related to elephants. After realizing that, it's difficult not to see the similarities between them and elephants when you look at them closer. And just go back on this. There's many other examples of vestigial organs. The appendix in humans and other things like this. These are structures that they've lost their function, but nonetheless they're still there. There's no real reason to. I guess they're in the process. We're in the process of losing things like that. All right. All right. Then we got molecular homologies. We share a lot of DNA with primates. With many of the great apes, we share a lot of our DNA with them. And as you go further out, we begin having less and less similarities with DNA. And also, with protein. So we're looking at this map over here. We're looking at how similar our hemoglobins are across these various species. All right, so we have 138 similar amino acids on our hemoglobin as a rhesus monkey, and 114 as a dog. We have 101 similar amino acids in hemoglobin as birds, and 21 as a lamprey. So not all that close to lamprey, but the idea is that you can kind of tell the amount of time that's passed based off of how our hemoglobin sequence or structure has changed. So one way to really look at the tree of life is a big family tree, right? And that's a great example here. When we talk about evolution, we talk about trees, we talk about clades, how the different species are related to each other. And so evolutionary trees are hypotheses about the relationships among different species or different groups. Homologies form nested patterns in evolutionary trees, and evolutionary trees can be made using different types of data. For example, anatomical and DNA sequence. A lot of times, DNA sequences, the primary driver in what we think of being the correct trees. All right, so example of one here. Hawks and other birds are more closely related to ostriches than they are to crocodiles, and crocodiles are more closely related to lizards than they are to mammals. And right, and so we begin to see these relationships forming here or displayed. The point of intersection are common ancestors. So if you want to go back to these kind of these trunks or these branches coming out, that's where there's a common ancestor between. For instance, you see this little purple line here in front of the sixth where it says feathers. So that's our ostriches and our hawks and other birds are the common ancestor for them. And after 5, we have a common ancestor for crocodiles and the birds. And after 4, there's a common ancestor between the lizards and the crocodiles and the birds. All right. Now there's something else called convergent evolution, and this is where life has essentially solved the same problem or came to the same solution. It's come to the same solution, different paths. Birds and pterosaur and bats or fruit bats, they're not greatly related. They're not in the same evolutionary lineup of each other. The bird and the dinosaur might be a little bit closer. Fun fact that birds, they're the last avis. It's the last living group of dinosaurs. So, when you see a chicken walking around, you're looking at a descendant of a dinosaur. All right, so, but these have solved the problem of flight very similar ways. They developing wings and other structures like that, but they did not develop it in the down the same lines of evolution. So they came to the solution in different paths. We see this in plants too. So we talked about the CAM plants and C4 plants. So it turns out there's a lot of C4 plants, the type of photosynthesis out there, but many of them came to, they developed C4, that technology, if you will, those traits independently. Cold-blooded, or warm-blooded, I should say, warm-blooded is also another trait that has evolved separately in different species, in mammals, and dinosaurs and birds. All right. And then we got analogous structures. So these are structures that are similar to each other, but are didn't evolve together. So they're solving the same problem. Again, they're solving the same problem, just like the convergent evolution. This is essentially convergent evolution. They're solving the same problem, but they have different paths. So in here we're looking at flippers, fins, wings, right? These are solving the exact same problem, but they didn't evolve from as a line from each other, right? Sharks didn't evolve into birds, and birds didn't evolve into dolphins, right? These are separate lineages. And then, fossil record, looking back, we can see both of these things. We can see homologous structures. We can see convergent evolution. When we're looking at some of these structures, and that's another example, or that gives some more evidence for evolution. And then we also can find basically what you might have seen on the missing link. We can find things where these transitional species. So, for instance, this particular organism is a transitional form between fish and tetrapod. All right, so biogeography, that's a study of the distribution of patterns of living organisms around the world. All right, so 200 million years ago, there was a supercontinent called Pangaea. Basically, if you just think about all of our continents squished together on the other side of the world, that's what Pangaea is. We've been slowly separating from Pangaea ever since, and we will probably meet back up at some point in the distant future on the other side of the planet as another supercontinent. So we can use this understanding to predict where species originated and locate fossil evidence. So it happens to be that we can find related fossils in different parts of the world that really only make sense once you realize that South America and Africa touched, and that Great Britain would have been sort of near the east coast of North America, and India was near Antarctica and that sort of thing. So it begins to make sense when you understand that these land masses were once contacted or in contact with each other. Then you have island environments, kind of like Galapagos. Um, Galapagos, both the Galapagos and Madagascar are isolated from mainland, and they haven't had a whole lot of introduction. There hasn't been a whole lot of um, migration of organisms from the island to mainland and vice versa. And you find that they have a lot of species that only exist on those locations. They've evolved in isolation from everything else. Marsupials in Australia. Australia has been basically separate from the rest of the world for a long time, and that's led to the evolution of things like marsupials and other organisms that are not found in other parts of the world. All right. So we're going to talk about the origin of species, how species come about, how we end up with different species. So macroevolution, that's evolution on a grand scale, formation of new species or groups of related species, results from speciation events and extinction. All right. So we have the accumulation of small changes in trait frequency results in new species and groups of many new species. So speciation is a conceptual bridge between micro and macroevolution. Basically, the evolution on the small scale and then evolution over a population size. So you might think this is kind of a silly question. What is a species? Well, a dog's a species and a cat's a species, and we know that cabbage is, different than celery and that sort of thing. It should be pretty simple to define what a species is, but in fact, it's actually quite difficult. And one thing you have to appreciate is that a penguin doesn't know that it's a penguin, right? It doesn't know that it's a different species from, say, a ostrich or a gorilla, for that matter. We as humans have attempted to make sense of all of these organisms we see around us, our neighbors, and we group them and try to organize them and put them in bins that make sense. And we've gotten better at it, but it's not always easy. So we'll go through some ideas of what it means to be a different species or to be the same species. So we say it's a group of related organisms that have common attributes. All right, it's pretty broad. Share same evolutionary history. All right, they're genetically similar, and they have similar morphological features. All right, so it is difficult, right? You look at these two birds up here. They're both yellow. They both have, dots on them. They both have black splotches near their necks. They look like they might be the same species, but they're not. So how do we identify them as being different species? Well, we could look at their genes. To have similar genes, we could look at their where they're located. We can look, do they breed with each other? There's a lot of ways that we can determine. And then on the other side of this, we look at us. We look at humans, and we're all the same species, but we have a lot of variation, right? We all hair color, skin color, facial feature, different facial features. So lots of, and even within a family, there's height differences and things like this. So it is difficult to define what a species means, right? All right, so you could say it's a population whose members have the potential to interbreed, all right? So it needs to be a naturally occurring population. They have to produce viable, fertile offspring. That means offspring that will live and then offspring that, when bred together, will produce more offspring. Um, and they cannot successfully interbreed with another species. All right. So gene flow occurs between populations of a species. So it says gene flow holds populations together. So in other words, basically, it's, if you look at these two groups, populations of bugs, we've got a brown beetle on one side, green beetle on the other. Maybe they're same species, maybe they're not. But the way that they remain the same species, as long as there's genes flowing between the two populations, they'll remain, in theory, the same species. They'll continue to interbreed and interact in that way. But the moment that they start, there no longer is gene flow in between the two, their evolutionary trees begin to diverge, their pathways start to go different directions, and they begin to become different populations, different species, right? Because they're no longer marching through time together and being selected together. All right, so one way that we can prevent genes from flowing from different populations is various forms of isolation, reproductive isolation. So there's two main forms of reproductive isolation. There's prezygotic, postzygotic. Prezygotic is basically before mating, before the zygote is formed. So before mating or fertilization event occurs. So there's habitat isolation. They live in different places. Temporal isolation. So maybe one is hibernating while the other is awake, or maybe they have different breeding cycles. Behavioral isolation. So we get an example of one of these birds here. Maybe they have different mating rituals, and so they're not going to be attracted to each other because of this. Mechanical isolation could be they just either because of different sizes or of different for whatever reasons, there's very different things that prevent each other from mating. A finch or a canary is not going to mate with an ostrich. That's one example there. And then there's a gametic isolation. In some cases, the gametes will not merge. The egg will not fertilize. The sperm will not fertilize a particular egg. Right. So, plenty of ways in which a zygote will not form. All right. So, habitat isolation. Species occupy different habitats, never meet to breed. Right. They need different breeding habitats to be successful, so they live in different areas, or they have different food needs. So basically, they're either living in different places, they, for whatever reason, they can't survive in the same place together, they like different foods. But this leads to them being different distinct populations, different breeding populations. They do not exchange genes. And because they're not exchanging genes, they're not locked together evolutionarily. Temporal isolation, we might see that species have different reproduction times, different reproduction seasons, right? Different breeding seasons, or they may be active during different times of the day. One might be nocturnal, one might be diurnal. Different reasons why two groups that live near each other may not come in contact with each other or may not mate with each other due to temporal behaviors. Behavioral isolation, largely this is like mating rituals. I think of bird calls, fireflies. Maybe the firefly doesn't do the right dance or doesn't flash its abdomen the right frequency or something like that, and they won't mate with each other. So behavioral isolation is a powerful form of isolation. Mechanical isolation. Perhaps they just can't mechanically mate. Genitalia are not compatible. There could be a size difference. We talked about like a finch and an ostrich. They're not gonna mate. All right, so those are mechanical means of isolation. So we're not gonna see any genes flowing between these organisms. And then gametic isolation. Egg and sperm may not be compatible. They may not be able to fertilize each other. So for an example, sea urchins have a recognition protein on the cell surface that facilitate fusion. They're not going to fertilize non-specific eggs. It's just not going to work, right? So gametes will not merge in that one. All right, so post-zygotic, these are isolation mechanisms that occur after mating or after basically the formation of offspring. So you could have a hybrid that is inviable, doesn't live, a hybrid that's sterile, it can't produce offspring, a hybrid breakdown, all right? Hybrid breakdown is where they're viable and fertile, but they have particular unfavorable traits that make it to where they're not going to be successful. So basically, they're not going to be fit, and they'll be selected against. All right. So we have some limitations to understand this fossil records, asexual reproducers, and hybrids. So what I mean by limitations for these, we find it difficult necessarily to group some fossils in the same species, right? We're looking at limited structures. We don't have DNA. We don't have that sort of thing. Where do we put some of these fossils? As far as speciation, trying to understand how to classify them. Asexual reproducers, they're not, so we don't have the mating criteria when we talk about these prokaryotes. So how do we necessarily determine them being the same species or different species? We obviously can't use their ability to mate as an example of that. We have to go by essentially just molecular and morphological and structural traits in that case. And then hybrids just kind of mess it up, right? Because are they the same species? Did they become a new species? You know, if they're viable, are they a different species from their parents? It's a difficult thing, right? So other definitions of species, morphological species based on structural features, it can be subjective. That's likely the way that species were determined in the past before we understood genetics very well. Ecological species concepts based on ecological niches, even in the presence of gene flow. So certain things like elephants, they may be able to mate with each other, but the two species of elephants, they don't live near each other, and so they're considered different species. And then phylogenetic species concept, smallest group of organisms that share a common ancestor, and that's probably, in my opinion, one of the more concrete ways to determine if something's a species or not. It's much more robust than the others. All right, so mechanisms of speciation. So what is speciation? That's where um, two, when a group of organisms, they become they become separate species. It's the creation of different species. So cladogenesis, the splitting of one species into two. All right, it occurs via two mechanisms: allopatric speciation and sympatric speciation. So allopatric speciation involves geographic isolation. So we look at the cartoon in figure A, we see the bluish green fish are completely separated and can interact with the goldfish. All right, and because of this, they're not sharing genes. Remember, the key thing about speciation, about different populations being different species, is whether or not you can have gene flow. All right, so when we're talking about speciation, we're talking about how you get different species. All right, now sympatric um, does not include geographic isolation, so we're looking at these, you know, the blue fish or the green fish and the goldfish. They are able to interact with each other. Right. So allopatric speciation, so that's speciation that occurs due to geographic isolation. So maybe they can't get to each other, and so therefore they can't swap genes, and they begin to diverge as two different lineages of organisms. So separations allow for interbreeding. So, for example, the Galapagos finches. All right, so mosquito fish, geographic isolation, live in separate ponds. Um, there's some genetic change, and evolution results in changes of alleles in populations due to different environments, results in morphological differences. Behavioral isolation, um, females mate with males of similar body type. Right? All right, so you have adaptive radiation. So you have a small population of organisms. They migrate into a geographically isolated area. And they split into different species to occupy different niches. Um, so essentially, one way to deal with competition is to find a niche and not having to compete with the other group, right? So this is one way they can find themselves into stable arrangements. All right, sympatric, that's speciation within the same geographic area. In plants, it results from polyploidy. That means having more um, chromosomes than they should. In animals, it results from sexual selection, so um, behavior, and maybe habitat differentiation. So again, it could be behavior. For example, these fruit flies, two appear to like red apples, and another two appear to like green apples, right? So maybe they're different species, or they become different species. All right, so polyploidy. Um, that results from an issue in meiosis. If you remember, meiosis is where we go through the process of making egg and sperm, or gametes, and we don't have um, the correct um, divvying up of chromosomes, and one cell ends up with more chromosomes than it's supposed to have. And what this tends to to um, to result in is that offspring can't reproduce. They can't interbreed with the previous group because um, in order to have a successful fertilization event, you've got to have the same number of chromosomes, right? And that's not typically possible in this one. Now, what can happen though is they can breed with themselves or with like very similar species. So you could end up with a new species pretty rapidly with polyploidy. So autopolyploidy originates from a single species due to cell division error. They cannot sexually reproduce with the original population. Speciation occurs in one generation, but they can self-pollinate. So you see this happening a lot in plants. And one important thing about polyploidy is that in some cases it gives that plant redundancy in genetic information. So it allows for further diversification because now it's got two copies of maybe a really important gene, and so now it can survive a mutation to that really important gene, and that mutation may confer a new characteristics that otherwise would not be possible because this mutation would have been fatal because it would have destroyed this necessary gene. But it's got two, so it can overcome that. Allopolyploidy, you have two species in a breed and produce hybrid. So this is basically where you've got two species with different chromosome numbers. So now the hybrid has an odd number of chromosomes and can't form. And typically with mammals, animals, they can't form successful or viable gametes, and they can't produce viable offspring because of that. And then with plants, this can result in viable offspring through vegetative propagation and then self-pollination as well. Sympatric speciation and sexual selection. Female preference for different traits can lead to different species. Color preference in female cichylids. Females of the same species choose males at different ends of the color spectrum. That's an interesting way of choosing. So they are basically they're isolating each other. They're sorting each other out through this preference that they have. Another one might be habitat differentiation. So these apple maggot flies, they would feed on hawthorns originally, but planting of apples in their environment changed that. So now you've got some that feed on the apples and some that feed on that hawthorns, and they've become reproductively isolated and becoming different species. Will likely become different species, possibly. Now, when you have geographic isolation, you can have places in between where these two populations meet, right? So you have maybe a little bit of gene flow between these two groups, and you have possibly some hybrids or some species in between that are visible. So you have an example here. You've got these different populations of deer, and they're able to meet up in some places along the mountains, through passes. And you've got, for example, these toads. They typically are isolated, but they have these regions where they're able to interact and share genes. Now, when you have these hybrid zones, you have three possible outcomes. You can have a reinforcement. So basically, the hybrid individuals, possibly they're not viable, or they're sterile, so they go nowhere, and so you have a continuation of the different species. Fusion, you could end up with a population where they basically they come back together. And so you see a despeciation and stability. They just march on. Maybe you got a population, a hybrid population in the middle that continues um, moving forward as well. So reinforcement. So natural selection strengthens prezygotic barriers. Barriers are stronger in sympatric species. Sympatric male flycatchers have different colorations. Females choose based on color. Right. Fusion, you have the barriers to reproduction across species are weak. Additional gene flow continues to weaken the barriers, and the two species slowly become similar to each other, and you've basically you end up with one population instead of having two separate populations. You have a despeciation. All right, stability. So hybrids may persist due to natural selection. Sometimes hybrids persist even though they are selected against, and that could be possibly due to a narrow range or zone. All right, so that might be an area where they're actually favorably selected against in the hybrid zone. All right, so the pace of evolution, you have two models, so the punctuated model and the gradual model. Punctuated model is just kind of happens. It's fast, and the gradual model is like it sounds, it's gradual. So in gradualism, each new species evolves gradually over time. Small changes accumulate over large time frames, results in larger changes. Punctuated equilibrium, evolution is more sporadic. Equilibrium species exist unchanged for long periods of time. Small changes accumulate, and then punctuated with short periods of rapid change occur. All right, so punctuated equilibrium, speed of change also dependent on generation time of species. So that's an important thing to recognize, is that in all types of evolution, the rate at which evolution can occur is going to be dependent upon the time between generations. So you think of elephants might have a lot of time between their offspring, between generations, but bacteria and viruses have very little time. So evolution happens really fast for bacteria. Evolution happens really slow for things like mammals and long-lived organisms. So rapid changes occur via several mechanisms: allopatric speciation and natural selection. Polyploidy creates new phenotypes. We can see where polyploidy in plants can produce a new species almost in one generation. Environmental shifts that drive gene pool in a particular generation. All right, so we might see a cataclysmic event that particularly shifts the population in one direction. All right, so take-home message. Speciation leads to groups of organisms that differ from their ancestors. Over time, some species grow in number, others decline or go extinct. Those are macroevolutionary events. All right, so we are at an hour and 8 minutes. So like I said, Charles Darwin was the man of the hour. So I'll see you in our next video. I apologize for the length of this one.